First Monday of each month. Laura is organizing this, and today we have Guido presenting about T cells, which is particularly interesting because the paper is about reduction of or prevention of the formation of fibrosis. And fibrosis is a very important factor for the genesis of arrhythmias, as you all know. So yeah, the tradition is that you can intervene at any moment with questions. If necessary, we can postpone them to later, but the idea is that you understand every step that Guido is taking in this very difficult paper that was published in Nature, I believe. So Guido, if you want to start off, please go ahead.
I hope you will enjoy this paper. I find it really interesting and inspiring. It's a little bit different from the usual literature that I offer you in the field of atrial fibrillation, aging, or metabolism, but I think it's going to be quite a nice adventure. It's a short report, so we're not going to see a huge amount of data, but we're going to see a little bit something a little bit complex, as it is the adaptive immune system. But you will see that in the last 3 slides, we will get back to cardiac function, and we will all enjoy the amazing results. So let me see. Okay, perfect. So as is usual, from when I present the journal club, I made some detailed presentation of the authors and the affiliated institutions, and we can see that this is a paper mostly conceived and carried on at the University of Pennsylvania in collaboration with I will just admit Fanny if I can. Yes. In collaboration with this company Acuitas Therapeutics, which is a quite interesting company. I found out that recently Pfizer has purchased an option for their lipid nanoparticles formulation to deliver some vaccines. And we will see that now in this technology that is presented in this paper, we all have it inside of us because we all received the COVID vaccine, which is based on this lipid nanoparticle technology. The journal is not Nature, but it's similar, it's Science. And of course, it's very well known. It's more of a commentary here. I usually put the mission or stated mission of the paper, of the journal, sorry, and Science really exploit the fact that it's a very well-known journal, and it just lists their major accomplished instead of putting their mission. But it's an interesting commentary. Now let's go to the topic of the paper. So as you can see from the title directly, we are talking about CAR T cells. So CAR is an acronym that stands for chimeric antigen receptor, and T cells are a part of the adaptive immune system, which is in charge of learning from antigen presenting cells how to either support other immune cells or to learn how to kill the target cell from which they get the antigen from antigen presenting cells. So as you can see in this nice picture, we will meet mostly 2 kinds of T cells: the CD4 positive cells, which are called helper T cells, and the CD8 positive cells, which are called killer T cells. And this is just an antigen presenting cells. It's a dendritic cells, but it's not important. There are many antigen presenting cells, such as in the heart. One of the most important is macrophages. So these cells are pretty much well known in the field of oncology, as you can transfect them with a viral carrier that is modifying the cell in order to recognize a certain type of cancer cell, which is in the best case scenario specific to the patient harboring the cancer. And the idea, as you can see in this scheme, is to isolate T cells from the host and activate them, so to render them immunocompetent, and transduce them with a viral vector, which in this case is lentiviral. It's called lentiviral CAR construct. Then you can expand them, you can isolate them, and you can re-administer them to the patient in order to target the specific cancer cells from which the patient is affected. And you can see this more details about how to use CAR T cells in this review that I put you the DOI of. But now in the field of CAR T cells for application outside of cancer, the authors highlight some important drawbacks. One thing is that a viral genetic modification, as some of you may know, is a permanent modification of the cellular genotype. And once these T cells get in presence of the antigen, they can divide more than 7 to 8 times. And this is in the presence of the antigen. This can be this number of divisions can be even increased. So this is very good for cancer therapy, because we want to get rid of all the cancer cells. But in certain conditions, such as in cardiac fibrosis, we might need to target a cell type that it's contributing to a pathological phenotype, but it's essential for other processes. And in case of cardiac fibroblasts that might get activated, they are well known to be essential for wound healing. So if you are killing activated cardiac, if you are killing myofibroblasts, you have troubles healing wounds while improving your cardiac phenotype. So the question of this short report is the one that I put in the last rectangle, is how can we make a temporary and hopefully a tightly controlled therapy targeted to an essential cell type, so not to a neoplastic cell type, that can contribute in a certain condition to a pathophyenotype. And here in this slide is presented the approach that the authors will use throughout the paper. As you can see, there is a lipid nanoparticle that is carrying messenger RNA. And this lipid nanoparticle has a decoration of antibodies on its shell that is targeted to a cluster of differentiation 5CD5, which is found in T cells, but is not it's not determining its function. So if we interact with this cluster of differentiation, we're not going to alter the function of T cells. As some of you might know,
Guido.
Please.
Can I ask a question?
Absolutely.
I wonder, what is the difference between liposomes? Is that the same?
No, no. It's now they are known with these lipid nanoparticles, but they are liposomes.
Oh, they are not. Okay.
Exactly right. And is actually the integration and the mechanism, it's typical of liposomes. So there is an endocytosis, an endosome escape, because they are made of phospholipids, and then they burst out their carrier, which in this case is a messenger RNA, and this is temporarily translated to express a specific protein on the membrane, which in this case is a fibroblast activation protein, chimeric antigen receptor, which will recognize this surface protein in activated fibroblasts. So differentiated kind of fibroblast, which is responsible for wound repair and progressive interstitial fibrosis. And in general, also stiffening, tissue stiffening during aging. I put here for your convenience kind of a template of the kind of mRNA carrier that we will find. So we will see that there are the types are made in this following this template. So first we will have the target on the lymphocyte. So in this case, it's CD5. We will have the type of carrier. So we might have lipid nanoparticle. We will have also retroviral vector as a control. And then we will have the mRNA cargo. So we might have a FAP-CAR, like in the final carrier that wants to be proven effective. But we will also have some pre-recombinase and GFP as controls. Because the field of liposomes is and lipid nanoparticles is fairly old, it is at least 30 years of age, and the technology of mRNA is even older, of course, it's almost 6 years of age. But it's a very complex world in terms of how to create these nanoparticles and how to make them biocompatible depending on the application. I put you this very interesting review if you want to read it afterwards. And then now we are moving to the results of the paper. So the first things that the authors want to demonstrate is that if you provide this genetic vector to freshly isolated T cells, then these cells will express the genetic cargo that they are transfected, temporarily translating in this case. So here you can find some details about the experiments. I'm not expecting anybody to be familiar with flow cytometry, but you can see here the color code of the different vectors that are used. And what they want to see in these results is that if we transfect these freshly isolated and activated murine T cells for 48 hours with a CD5 LNP GFP, so that will express only green fluorescent proteins when it's transfected in CD positive T cells, then we have an 81% of cells that are successfully expressing GFP and an 81.4% of CD5 positive cells that are expressing FAP-CAR when they are transfected with this to express this antigen. This is much more clear in this bar graph.
Guido, may I have a question?
Yes, of course.
Guido, so with these cells and the nanoparticles, so nanoparticles are like made or like tailored to the mice, like to individual mice, or are these some?
No, the aim is that the aim of this paper is that you will not isolate cells from the individual mouse in this case before genetically modify them. The aim is to modify them in vivo.
Mm-hmm. Okay.
So a priori, you have to choose a marker that is expressed in your target cell that will not interfere with their ability to functionally perform. Okay. So in this case is this CD5 surface marker that is targeted by this lipid nanoparticle.
Okay, good, thanks.
You're welcome. So so to assess the specificity of the expression of FAP-CAR when T cells are exposed to different kind of genetic vectors, they have compared the expression of FAP-CAR positive T cells, and you can see in this graph that in vitro is very specific. This IgG is the antibody control for the antibody. So it's just a specific immunoglobulin which will specifically attach to T cells in this case, because we are talking about isolated T cells. And so you can see that the expression is very specific to the carrier that wants to translate FAP-CAR. Much more interesting, because these results were already published by this group. Much more interesting is that this formulation is indeed transient. So you can see that in time, the percentage of isolated T cells that express FAP-CAR is decreasing in time, as cells are either dividing or are dying. So this is already a good preliminary result of the transitory nature of this genetic modification. Then, of course, they need to check whether this could work as cytotoxic T cells, because we are planning to kill a specific kind of cell. And following the same set of genetic vectors, they have transfected this fibroblast activation protein on human embryonic kidney cells, which some of you might know. It's a model dummy cell where we can express a protein of interest, and then we can, in this case, target it with genetically modified T cells. So the killing efficiency in these target cells is really specific when specifically higher when T cells are expressing this FAP-CAR antigen receptor. Not surprisingly, the killing efficiency is higher with another kind of genetic modification, which is the retroviral one, which, as I explained to you in the introduction, is permanent, is much more stable, and therefore has a higher killing efficiency than the transitory one that the authors want to demonstrate as functional. There's not a lot of replicates, but they do show, and I'm quite astonished by the fact that there are no statistical comparisons. This is also visible in the supplementary figures, like in this bar graph on the right. They want to prove that not only it was a good way to target FAP expressing cells, but that this was also dose dependent. So if we increase the number of modified T cells that enter in contact with modified HEK cells, then of course the percentage of killing is higher.
It is very remarkable. If you go back
Please.
a bit in the left panel,
Right.
you see,
Right.
and the black bar, you see individual data points, but not in the bar itself. So they're masked, and
Yes.
probably the spread is very high there, I suspect.
You think here I see only 3. I think there are only 3 replicates.
Oh, really? Well, I don't see the
only
third one. Oh, okay.
It's here behind the standard error of mean.
Okay. I don't see it. Okay.
Yeah, I can see it on my screen.
It made me suspicious.
There is one. But yeah, of course, it's like 3 replicates. So then they passed to do to prove this specificity and transitory genetic modification in vivo. So they took, I could not find the strain of the mouse, but I know it's a wild type in the paper. And so as I was mentioning, they play a lot with the kind of gene that they are transfecting in these animals. So follow me. Here we have a lipid nanoparticle, which is decorated with an IgG, a generic IgG. So it's like a control for this antibody. This one is targeted to T cells, and when it's transfected, it's not expressing this FAP-CAR protein, but it's expressing luciferase. So we can see in the organs the accumulation of these transfected cells or of the genetic vector via luminescence. So if we look at this grid here, we can observe that of course, like in naive samples, which means that are transfected just with that are injected just with vehicles. So this is assessed, I think, 48 hours after injection. So in the naive carrier, so sorry, this is not saline, it's just an empty lipid nanoparticle. There is no specific fluorescence that we can see here, either in either explanted organ. If we look at the control for the antibody, this IgG-LNP luciferase, we can see that the accumulation of this genetic carrier is mostly in the liver. And the author says, and it's partly true, that this is expected because this lipid nanoparticles, these liposomes, are mostly cleared through the liver. So this doesn't mean that there are transfected T cells, that these transfected T cells per se. This might just be a specific cells that are accumulated in the liver. When we transfect specifically to CD5 cells, indeed we see a much more increased signal in the liver, and we see a specific signal in the spleen, which is the organ which is deputed to filtering immune cells and blood cells in general. So in vivo, it does seem that this approach is specific to reprogram local T cells, the T cells of the host. But of course, one could say, you are looking at a luciferase, could be highly aspecific. What are you doing? So they used a reporter mouse, which is this strain here. It's AI6 Cree reporter. And I don't know how many of you are familiar with Cree-Lox recombinase strategy, but in this case, this mouse has, in its genome, has this kind of genetic sequence that blocks the expression of a fluorescent protein, which in this case is ZS green. When it's the Cree recombinase is entering in contact with this site, this sequence is cut, and now the fluorescent reporter can be expressed. What does it mean? That if the T cells are transfected, they will shine in green. And as expected, otherwise it will not be a result, we can see that indeed the targeted genetic vector specifically increases the expression of these fluorescent markers. We can see here why it's a research report there are just 2 points. So this CD9, this CD3 negative are non-T cells, immune cells. So it's at least I would say 70% is specific to T cells, and it's specific to both kind of T cells. So helper cells and killer cells. So in this case, they have confirmed that they can reprogram using this targeted approach T cells and only immune T cells in vivo. And now we get to our pathological model. So it's a fairly common model. It's the angiotensin-phenylephrine injury model that produces cardiac fibrosis in the span of 1 to 3 weeks. So these mice are implanted with an osmotic pump, which is giving constantly this dosage of angiotensin and phenylephrine. After 1 week, fibrosis is canonically installed, and they are injecting them with this genetic vector. And now they say that at the end they will analyze the cardiac function at 3 weeks, but now they are looking at isolated immune cells from the spleen after 48 hours. So in this flow cytometry experiments, they show that roughly 25% of T cells that were isolated from the spleen after 2 days were expressing this FAP-CAR antigen receptor, and if you transfect them with GFP expression, it's around 17%. And again, in vivo, as we saw in vitro, the FAP-CAR expression is really specific to mice who have been injected with this CD5 LNP FAP-CAR genetic vector. Yeah, and again, like it's a little bit close, in average, close to 25%, which is not bad. And as we saw before with the Cre recombinase, this affects both kind of T cells, both CD4 positive and CD8 positive. And what is interesting is that most of the transfected cells that express FAP-CAR are helper cells. So this is a little interesting, which means by means that the mechanism through which this genetic modification acts as an amelioration of fibrosis might not be directly cell killing. But the report doesn't go into this detail. One other function to test for a competent T cell is to observe this. I found out this fairly little known process, which is called thrombocytosis, where a T cell will bite a bit of the membrane of the target cell, and it will then process it, and it will present it on its own membrane. I didn't go too much into detail for this journal club, but it's linked to cell death. It's linked to adaptation of the immune response again. But what the authors are planning to do is to transfect HEK cells again with fibroblast activation protein, so to make it again a target cell for this genetically modified T cells. And at the same time, they're expressing this RFP which is, you can see it here in pink. And in this video, you can see these are the HEK cells at the center. These are little ones are the T cells modified. And you can see that when the T cells enter in contact with these HEK cells, they are appearing some puncta inside this T cell in contact, which it's a sign of this trogocytosis in progress. So in order to prove this further in vivo, they performed the same injections that we saw before in control mice and injured mice with this angiotensin 2 phenylephrine insult. And if we look at the micrograph of this fluorescent fibroblast activation protein, we can see this is a micrograph of the spleen. This is the pulp of the spleen, which is responsible for the filtration properties of the spleen. And so we are expecting immune cells to accumulate here in this area. And we can see that only in injured mice, so that present these activated fibroblasts, that present cells that have been genetically modified to recognize this antigen. We can see that there are bits of this protein inside the pulp of the spleen of these mice. And here they show that indeed these T cells in this case are expressing GFP, and so these are T cells that are passing through the spleen, and they present this puncta of fibroblast activation protein, which is similar to the video that we saw before. It's proving that this T cells are biting a little bit of the membrane out of these target cells. And they also prove it in the injured mouse, but with the final formulation and with its control. So they are looking at surface markers here. So this CD3 plus is any T cell. We can see that in the injured mouse with the specific genetic vector, indeed there are these T cells with this puncta of bit of fibroblast activation protein.
Yeah. So, Guido, the cells that are subject to this trogocytosis, they don't die then, or eventually they do?
They do. It seems to be the mechanism through which, after a while, the cell dies.
Okay.
It's another mechanism other than phagocytosis.
Okay.
As far as I could see from the literature. Of course, I am not far from being an immunologist, so I research only the strict necessary for you to navigate through these results. And now we can go to what we understand, appreciate, and we can absolutely enjoy, which is the effect on the cardiac function. So here we go back to this little schema that shows the injection time and the analysis time. So here we are. The injection is performed 1 week after beginning of the injury, and then the final analysis is performed 3 weeks after the beginning of the injury, so 2 weeks after the injection. So we observed 3 groups. Injection with saline, pumping with saline and injection with saline, pumping with the injuring solution and injection with saline, and so this is a sham and this is a control for the genetic vector. And then we have the group of interest, so injured mouse treated with this CD-specific cardiac fibroblast targeting genetic vector. This is echocardiography. You can deduce, suppose from the parameters that are explained. So here they have enough numbers, replicates to do some statistics. So these animals that are injured, they develop an expected phenotype if they are not treated with this genetic vector. So they develop contractile dysfunction, which is observed in increased diastolic volume, increased dense systolic volume, and as we can expect, reduced ejection fraction, reduced global longitudinal strain, left ventricle mass index as calculated, and also valvular dysfunction with increased E to E prime ratio. Of course, what is interesting, and I have to go back for this, is that when you are injecting these injured mice with this genetic vector, so these mice are expressing reprogrammed cytotoxic and helper T cells, then this injured phenotype is disappearing. It's reverting back to the sham phenotype in basically all parameters. So this treatment seems to prevent the installation of the pathophenotype. And the last slide, it's you can see an histology of cardiac fibrosis in the ventricles, and as observed before, we can see that indeed this seems to be this recovery of functions seems to be linked to a reduced fibrosis, which either is not installed or because they are waiting 1 week before injection of this genetic vector, it's actively reduced by CAR T cells targeting activated fibroblast. So the general conclusion of this short report is that using lipid nanoparticles, which deliver messenger RNA, this hyping words, messenger RNA makes it possible to transiently engineer CAR T cells to alleviate the contractile dysfunction that is correlated with cardiac fibrosis. And this is the end of the presentation. I would like to thank you for your attention. I hope that it was as clear as possible.
Well, thank you, Guido. That was an excellent presentation and on a very difficult topic. And I think you rightly assume that this is very important because, as I said before, fibrosis is so important for arrhythmogenesis. So the obvious question is, how are we going to implement this in arrhythmia studies? Do you have any idea, or any other of the audience?
If I may, like the first thing that came to my mind, of course, me dealing with aging, is target senescent cells.
Yeah. Because
they express a very typical surface marker.
Yeah.
And
but you know, atrial fibrillation, at least persistent atrial fibrillation, is associated with atrial fibrosis.
Indeed.
And you know, Brugada syndrome and early repolarization syndrome are now thought to be associated with very subtle forms of fibrosis as well. Yeah, in heart failure,
yeah,
arrhythmogenesis based on fibrosis is very obvious. So I think I see a lot of potential targets for this strategy.
This is why I thought it was relevant to get out of my comfort zone.
Yeah,
explain something like this in immune therapies.
Yeah. If you go back one slide,
please. Yes.
you see the E over E prime ratio, which is a echocardiographic measure of diastolic dysfunction,
Mhm.
and it's associated with usually, at least that's what they think, but with a poor relaxation of the muscle if it's increased. And apparently this is normalized as well. So it's a very powerful tool. Although I do have a question that the normal value in humans is below 15, I think, or 14. So how does that work with mice?
No clue. Frankly, not an expert of echocardiography in rodents.
Yeah.
But I mean, in terms of comparison, I mean, for the scope of the paper,
Yeah,
it's pushing on novelty.
Yeah. Very nice.
Guido, I have a question.
Please.
Just to kind of interpret the results for me. If you go to the final slide with the fibrosis.
Yes.
So basically what we are looking at is on the very left side, there is the control, like the control.
Mm-hmm.
In the middle, these are mice in which the fibrosis was being induced for 3 weeks, right?
In all of them, except for the saline, which is the real.
Okay,
control.
yeah.
In all of them, the injection of these profibrotic molecules is carried on for 3 weeks.
For 3 weeks, but okay, but in the right one, the profibrotic molecules were administered for 1 week, and then on top of that, for the last 2 weeks, there was this treatment, right?
But there is only one injection.
One injection.
The idea is to give one injection
Mm-hmm.
that will transiently reprogram the T cells of the host,
Mm-hmm.
recognize activated fibroblasts, which express this fibroblast activation protein.
I see. No, because like what I'm wondering, like how to interpret the difference between the two groups. Like, does the treatment they propose, does it reverse the fibrosis, or does it just stop the progression of fibrosis? Because this picture can give an impression that it's kind of reversing the fibrosis, but it's, as I understand it, it just kind of stops the progression, no?
Yeah,
Yes,
it prevents it.
Yeah. The issue with this model, which is quite stated quite a lot in the report, is that in this model, after 1 week, there is already interstitial fibrosis that you can detect.
Mm-hmm.
So the claim is that it does both. It reverts, like it suppresses fibrosis to a certain extent and prevents the installation of more fibrosis. Yes.
Mm-hmm. But they don't have any evidence for that. It's that's an assumption, I suppose.
No, this is why I put like it's a correlative observation, which is definitely powerful.
Yeah.
The only thing that we can trust really is the data from the echo, and we can assume that is indeed driven by an antifibrotic mechanism.
Yeah, because like what I'm missing a little bit in this figure is like this 1 week time point. Like, how does the fibrosis look there? If it's similar to the third group, or if it's more similar to the middle picture. That was just my curiosity.
This we don't know, and it's not even in the supplements.
But it's a good suggestion, because that would allow you to differentiate between prevention or reduction.
Right. If you can see, the phenotype is in the middle of the two, at least, the quantification is in the middle of the two groups. So we can presume that, like really doing an educated guess, that it's mainly preventing further fibrosis.
Mm-hmm. Yeah, that's what I would guess as well.
Yeah.
Thanks.
For an arrhythmia, from an arrhythmia point of view, the middle section is very arrhythmogenic, because you see these isolated bundles, almost like in myocardial infarction. The yellow bundles that are isolated, they are a substrate for reentrant arrhythmias and activation delay.
Mm-hmm.
Yeah. very nice.
It's definitely a technology worth exploring. If
Yeah.
anything, because we all have it in our blood now.
Yeah.
I mean, the ones who had the Pfizer vaccine, but I think the Moderna one was also based on lipid nanoparticles.
Yeah. Okay. Any more comments? Fanny, how are you? Do you have any comments?
Hello?
Hello.
Yeah.
Yeah.
Yeah, I'm fine. And you?
I'm okay.
No, it was very complicated. I was just wondering if they look, so if there is a remove of the fibrotic cells, there is a production of new cardiomyocytes or to replace these cells?
Ah, yeah. Interesting question, but I don't think the cardiomyocytes will replace it.
I know.
Yeah.
And mice are really not a good model for cardiac regeneration because they have a lot of hematopoietic stem cells that can resemble kind of proto-contractile cells. So it's really a bad model for that.
Yeah, but this is a good suggestion because fibrosis, like in the middle panel, is based on apoptosis. So there's a reduction of number of cells which are not replaced. So I think the main mechanism is prevention rather than reduction.
In an infarct, you have a lot of fibrosis. So what will happen if we remove?
You wouldn't use it on an infarct.
And we can see that the valves are very red on the image. So this treatment can alter the valve.
Oh, but this is the Van Gieson staining. It's staining collagen. So any kind of collagen. So this might be like collagen 1. This might be collagen 4 on the valves. The Van Gieson staining is just staining collagen in red.
Yeah.
So it's not cell specific.
Okay.
Okay. Well, thank you very much.